Well, hello everyone. Welcome to tonight's event titled A Frozen Sea in Flux, Understanding Changing Arctic Ocean Ecosystems Through Art and Science. Thank you so much for being here tonight. We really appreciate it. My name is Evan Portier, and I'm an educator here at the U.S. National Science Foundation, National Center for Atmospheric Research, also known as NSF NCAR for short. As many of you might know, NSF NCAR is a world-leading organization dedicated to understanding Earth system science, including weather, the sun, and our atmosphere, and how all of these systems are important to build national resilience. Now, before we start our presentation, I have a few housekeeping things that we need to go over. First and foremost, we have a couple of questions by our presenters through the Slido platform. So for both our in-person and online audience, if you don't mind and you haven't had a chance to go to slido.com and type in Explorer Series, we also have a QR code that will pop up in just a moment that you can scan. On this platform, you'll be able to answer a couple questions for us. Speaking of questions, if you have, and there's that QR code right there. Speaking of questions, if you have questions, I ask that you please save them to the end of the presentation. We'll have some opportunity to ask our presenters questions, both online and in person. For our audience that is online, if you scroll down on the Slido platform, you should see a green button that says Join Event. Please click that, and you'll see a Q&A tab where you can ask questions. And as always, our NSF NCAR Explorer Series is being recorded, so any folks that weren't able to attend tonight can go and watch the recording as well. You are able to scan it, and if not, don't worry about it just yet here. So with that, I would like to introduce our two speakers tonight. We have Anna Lindemann, and Courtney Payne. Anna Lindemann calls herself an Evo-Devo artist. Her work as a composer, animator, and performer explores the field of evolutionary developmental biology, also known as Evo-Devo. She is an associate professor in the Digital Media and Design Department at the University of Connecticut, where she has pioneered courses integrating art and science. Courtney Payne is a postdoctoral researcher at the University of Colorado Boulder and NSF NCAR, having completing her PhD in Earth System Science at Stanford University. Her research focuses on primary production in the Arctic Ocean, where she uses models, satellite remote sensing, and field observations to study changes in the timing and magnitude of phytoplankton blooms and the impacts of primary production on Arctic ecosystems. And with that, I would like to introduce our two speakers up to the podium, and we'll go ahead and present our first question from Slido as a word cloud. Why don't you give them a round of applause?
If we can take a moment to process that polar bears is here, big and center, and we'll come back to that. And if you want to come back to our slideshow, we can take it from there. So this talk came about as a result of an animation called Arctic Shifts that was recently launched here at the NSF NCAR Mesa Lab Visitor Center. In this animation, scientific climate simulations and speculative storytelling combine to reveal the connections between our human activity and the ecosystem and environmental changes that are occurring in the Arctic. This is the region of our planet that is warming most rapidly due to climate change. So one of the things that we're thinking about with this animation and this evening is how are we all connected to the Arctic? And in thinking about our planet's complex interconnections, how does the Arctic matter to all of us, matter to you? So during this talk, we're going to introduce ourselves and this animation. We'll talk about the process of combining art and science in the animation. Courtney's going to talk about the science behind the animation. I'll share a little bit about the process of making the animation and music, and we'll end by playing more of the animation. This animation started when my friend Alice Duvivier, who's here in the audience, who is a scientist here at NCAR, when Alice and I started talking about collaborating on a project. And here's a picture of Alice and me when Alice hosted me as an official visitor at NCAR back in 2021. This project brings together our very different backgrounds, so I'm going to briefly introduce us. Alice is a polar climate modeling scientist. She's conducted some field research. Here she is at Sea Ice Camp in Alaska and taking ice core samples. But most of the time, Alice works with climate models to understand the polar regions. She uses the Community Earth System Model, CESM. Many of you in this crowd are experts on this, but for those of you who aren't, the Earth system includes the land, oceans, ice, and atmosphere of planet Earth. And the Community Earth System Model is a global climate model that provides state-of-the-art computer simulations of the Earth system in the past, present, and future. On the left is a picture of Alice in front of some of the supercomputers used to run the climate simulations. And a lot of Alice's work leads to scientific publications like the one excerpted here. In the figure excerpted here from this article by Alice, we see the decrease in Arctic sea ice volume over time for both reference and computer models, including some CESM configurations. My own interests come from a different direction of studying biology. My interests growing up included making things about nature. Here is my jellyfish Halloween costume when I was 12 and a piano piece that I wrote about bugs from when I was 12. When I went to college, I studied biology. I studied bird calls in Indonesia, and I worked in an Evo-Devo lab headed by Dr. Antonia Montero at Yale. The lab studies the evolution and development of butterfly wing patterns. And in working in this lab, I was really swept away by the stories that the field of Evo-Devo is uncovering. And I wanted to share these stories more widely. I now call myself an Evo-Devo artist. I bring together animation, music, and performance to explore evolutionary and developmental biology. On the left are some stills from an animated short called Beetle Bluffs that was shot in the Museum of Comparative Zoology at Harvard. And on the right is an art science performance called The Colony, which I had the pleasure of creating with Emma Komosrowski, who's also here in the audience. And this is a show about the evolution of social life and communication in both ants and humans. So Arctic Shifts emerges from Alice's background studying the Earth system and my own background developing artworks related to biological systems. With this project, we set out to illuminate the interconnections between the ecosystem and the Earth system, and our focus was on illuminating these interconnections in the changing Arctic. So many of us may think of polar bears when it comes to organisms that are being affected by climate change in the Arctic, evidenced by the Slido poll, where polar bears was really big in the results. One of our choices with this animation was to focus on organisms that are often overlooked. And for us, that is looking at the base of the food chain to the green speck all the way on the left, and that is phytoplankton. And I'm going to turn it over to Courtney to talk about phytoplankton.
Yeah, so phytoplankton are not what you came here to learn about, but you're going to be forced into it now anyway. They're these tiny microscopic organisms. here's a bunch of images of individual cells that are all phytoplankton. And I think they're really important for quite a few reasons. One is that they're the plants that largely do all the photosynthesis in the ocean. So they're the base of kind of the ocean system in a sense. They take sunlight and they convert it, along with CO2, into sugars. And they, as a byproduct, produce oxygen. And actually, phytoplankton produce half of the oxygen we breathe. So every other breath you take, you can be grateful to a phytoplankton for that. Phytoplankton, when I think about phytoplankton, I don't typically think about those individual little cells. I think about an image more like this. So this is a big phytoplankton bloom off the coast of Norway. And just like with land plants, phytoplankton bloom. They have a period where they start growing really rapidly. And we can see that from space. So this is, all these big green swirls are phytoplankton going crazy in the Arctic here. You'll notice the swirls as well because phytoplankton are drifting freely with the ocean currents. So they just move with the flow. As I mentioned, I think phytoplankton are important. I think you'll think they're important in a moment as well. This is an Arctic food web. And at the base of that food web is phytoplankton. So everything else that we might care about on this image, polar bears, humans who live in the Arctic, fundamentally relies on phytoplankton. And even after they die, phytoplankton are quite important. When they die, they sink down to the bottom of the ocean, and there they can feed clams or sea stars that live in the sediments. But they also can take that carbon that they sucked up in the surface of the ocean, and they can sequester it, kind of keep it down in the sediments for really long time scales, thousands of years. As you can probably imagine, I didn't start off my life really interested in phytoplankton. I was just a person who was really interested in spending time outside. And it wasn't until I was a college student that I kind of fell into studying phytoplankton and oceanography. I took an oceanography class to get out of a science requirement as a college student. And I was also rowing at the same time, and I was rowing in the ocean. And so every day I was breaking sea ice with my oars or watching a phytoplankton bloom and turn the waters around us green. And I think being able to kind of understand the Earth system really excited me every day at 5 in the morning. I also then watched, as a sophomore in college, a film, Chasing Ice, which is a film made by James Bailog, a cinematographer here in Boulder, that attempts to explain why the polar regions are so captivating and why they're so important to capture because of the big changes happening there. So this film really inspired me to specifically study polar oceanography, and I ended up getting a National Science Foundation funded research experience for undergrads, which brought me to the Arctic as an undergrad student. So I got to study phytoplankton and changes in phytoplankton off the coast of Norway. We're in this place where glaciers were calving and really changing the environment. I subsequently went and got my PhD, and for the most part, I sat next to a computer, staring at it for many hours in a basement. But every once in a while, I got to go and do field work in the American Arctic, and it was, it really inspired me to continue studying the system. So we had a question for you all. We were curious to know what you thought is a way that you think that the Arctic environment is changing. Let's see what you all came up with. Okay, so I see a lot of people saying that sunlight is changing in the Arctic. That's certainly true. Thawing permafrost, we're not going to talk about that too much because it doesn't super directly impact the ocean. But I saw a lot of warming and melting and temperature. Those were things we're all going to discuss today. So I'll talk to you now about some of the ways that I'm thinking about the Arctic and why it's changing. And I think these, again, are the main reasons I'm motivated to study the Arctic. So I'm going to start by showing you this temperature anomaly figure. And an anomaly means that you look at the change in temperature relative to a baseline. And so in this figure I'm going to show you, red means that the conditions at present are warmer than that, relative to that baseline, while blue means it's colder. And here's a map of the whole Earth. And we can certainly see that the whole Earth is getting warmer. But the thing that really sticks out to me about this image is the Arctic, this part up at the top of the world, which is, of course, warming at a much more rapid rate than we see across the rest of the globe. It's actually warming about 4 times the globally averaged rate. And that warming has big consequences. This is an image of sea ice in the Arctic, this relatively thin layer of ice that sits on top of the ocean. This is taken in the 1890s during an early polar explorer trip. And you can see in this kind of ice pressure ridge that's formed, massive chunks of ice. They're thick. They're really highly reflective. Like you can imagine that not a ton of ice would get through this very white surface. And ice like this was relatively extensive. It went down pretty far south. Ice in the present Arctic looks really different. It is thinner. It is younger, which has some pretty big consequences. It certainly lets a lot more light into the surface of the ocean, both through leads and also the thinning. And it's less extensive. It doesn't go as far south as it once did. And all of that has some pretty big consequences for the ecosystem. So I'm now going to turn it over back to Anna.
Is it changing the salinity?
We'll get to that. Yeah.
So, this brings up that we've talked about, Courtney talked about the thinning ice, but that's just one of the many changes happening. This is immediately, as soon as you start to think about what are the effects of the thinning ice, you start to understand that this is a really complex system. So coming back to the animation that I was creating with Alice, one of the questions that we sought to understand was how can we communicate changes happening to a complex system in an accessible way? And one of our responses to this question involves combining art and science. I think one of the greatest challenges with the subject of Arctic shifts and with our own species' capacity to understand and address climate change is the challenge of wrapping our minds around processes that are beyond the human scale. So that means things that are microscopic. They're too small for us to see, like the phytoplankton that's way at the base of the food chain transformed inside the stomach of the polar bear. Or that means things that are too large for us to see. So things that are happening at a global scale, like the temperature anomalies. And understanding what are the connections between the microscopic and the macroscopic. In Arctic shifts, I leveraged different artistic media to try and communicate those connections. So with Arctic shifts, I thought about the power of the medium of animation to connect microscopic and macroscopic processes. On the left, you can see a continuous zoom allows us to move between microscopic and macroscopic fluidly and to connect the phytoplankton to the Earth system. I also thought about diagrammatic design as a way to provide context at different spatial scales. And color design creates connections across scales. So the same green color is used when we see a microscopic view of phytoplankton, when we see the individual cells, and when we see a macroscopic view, when we see the swirls of the phytoplankton at the scale of planet Earth. I also thought about the power of sound to connect the microscopic and macroscopic processes. So each of the organisms and Earth system features in Arctic shifts has an associated unique timbre. As one example, most of the Arctic phytoplankton that are highlighted in the animation are diatoms, and these are creatures that have glass shells. So I use different sounds produced by glass to underscore the phytoplankton at both microscopic and macroscopic scales when we see and hear them in the animation. With Arctic shifts, we also really wanted to reach different kinds of audiences. We wanted different kinds of audiences to understand changes that are happening to the Earth system and ecosystem in the Arctic. And at the same time, we wanted to elicit an emotional reaction and be sensitive to feelings of climate anxiety that people are bringing with them. This led us to combine both diagrammatic explanatory animation with more emotive poetic animation. So as we were developing this animation, we developed it over many iterations, and we gained feedback from different kinds of audiences along the way. So one of the audiences that was important to gain feedback from was from scientists and experts in science education. Becca Hathaway, who's the director of UCAR Center for Science Education, was involved from the very beginning, from our very first pitch document and search for funding. And the explanatory direction of the project was really amplified through her feedback. We also met with scientists to share in progress work, and it was through their feedback that we honed in on the stories we wanted to tell, and also finessed the wording to make sure that it was accurate to the science. At the same time, we shared in-progress animation with middle school and high school students, and we asked the students to respond to questions about the science shown in the animations, and their responses to those questions led to us reworking a number of the aspects of the science visualizations. We also asked the students what feelings they had, and their responses ranged from wonder and sadness to guilt and determination. So this animation looks into the future. It asks, what happens to the food chain in the new Arctic? And the reality is the future has a lot of uncertainty to it. And one of the things we've decided to do with Arctic Shifts is to depict that uncertain future by embracing nonlinear narratives, by telling 3 different stories of possible Arctic futures. And these stories combine elements of scientific and artistic models of the future that consider how Arctic phytoplankton and the Arctic food chain more broadly will change in the new Arctic. One of the questions we asked all of you through Slido was, what is one factor that controls the growth of phytoplankton that might be considered when thinking about what will happen to phytoplankton in the future? And I'm going to turn it over to Courtney.
My work here is done. You already know exactly how phytoplankton are being affected. I see a couple interesting things. A lot of people have highlighted sunlight and sun, and then temperature also, and nutrients. So these are, as well as carbon dioxide, pollution, really interesting things. We're going to focus largely on sunlight, temperature, and nutrients today. I'll now talk to you a little bit about these 3 primary controls. So, light, nutrients, and temperature are all things that phytoplankton require to grow. When light increases, when nutrients increase, when temperature is higher, phytoplankton grow more rapidly. Anna and I are going to tell you 3 stories today, and they're fundamentally stories about the ways that these 3 environmental factors are changing. In order to tell those stories, I'm going to have to use a little bit of jargon, so I'll explain a couple things first. Today we'll talk a little bit about changes in phytoplankton biomass. And biomass is maybe a potentially scary word that just means the mass of biology, in this case phytoplankton. So it's a measure at a particular snapshot in time of how much phytoplankton there are. We'll also talk about bloom timing. And the same way that plants are accustomed to start to bloom on a particular date, phytoplankton have a start timing to their bloom, and that might be shifting as well. We'll also be talking about productivity. This is a measure of the net uptake of carbon through photosynthesis. While biomass is a snapshot in time of the amount of phytoplankton on a given day, productivity is kind of a measure of how much growth happened over a whole year or a month or a day. So it sounds very similar, but it's a very slightly different thing. And finally, we'll be talking about changes in species. This is something that Anna's animations really depict very nicely. As I talk you through the science, I'll be relying on a bunch of different studies. Some of those rely on computer modeling, others on satellite remote sensing. And also, I'll show you some results from these in-person investigations. These are largely done in boats in the Arctic. And each of these tools we have for understanding the world has some really big benefits and have some really big drawbacks. So we'll be discussing those a tiny bit at the end as well. So I'll start off with our first story, the story of light. And I think in the Arctic, this is fundamentally a story of scientific consensus as well as bloom timing. The Arctic is an environment that's highly controlled by light. Light is a very important factor. For months of the wintertime, depending on latitude, there's absolutely no sun at all. And even when the sun returns, sea ice and snow cover prevent that light from reaching the surface of the ocean for several months of the year. So when we typically think about phytoplankton in the Arctic, we think that the blooms don't really happen until around the time of sea ice breakup and retreat. And then you'll get this big, intense burst of primary production, this big bloom of phytoplankton that can feed the rest of the ecosystem. And of course, as we've kind of discussed, sea ice conditions are changing really substantially in the Arctic. We know sea ice is thinner. It retreats earlier and advances later at any given place. It doesn't extend as far south as it once did. And so that means that there are some really big impacts on phytoplankton. I was really interested to study how these blooms are changing as the environment changes. Here's an image of kind of an idealized phytoplankton bloom. I have day of the year down here on the x-axis, and the y-axis has phytoplankton biomass, how much phytoplankton there were on any given day. And you can see that they start off at a very low concentration. They increase. Maybe the bloom starts on some particular day. We could adjust that along this curve. The bloom peaks, and then it dies off. And this is kind of our classic idea of what a bloom looks like in the Arctic, one single intense burst of primary production. So I was curious to understand how much earlier the bloom will start. Because sea ice is getting lost, we know there's more light in the system, and the bloom will start earlier, but I think how much is not something that's well understood by science. And I was also curious to understand if biomass might be less concentrated in a single bloom in the future. This is kind of a more typical view we have of what blooms look like at slightly lower latitudes than in the Arctic. But there's maybe some ideas that biomass might, that phytoplankton blooms might look more like this double peak shape in the Arctic of the future. And this work I did using computer models. So I'll start off by talking about changes in the start timing of the bloom. For this study, I took the start timing in 2100, and I subtracted it from the start timing in 1970. So red indicates that the bloom starts later in 2100, and blue indicates it starts earlier. And not surprisingly, given I kind of gave you the answer already, in the Arctic, we largely see a bloom that's shifting earlier in the year. Pretty much everywhere in this map is blue. And just to orient you, here is Alaska and Greenland and Russia over here. There are some places where there's no significant change over time, these kind of polka dotty places, but for the most part, everywhere's kind of getting earlier. On average, the phytoplankton bloom starts a month earlier by 2100 than it does at present. That's even, it's even earlier at certain places near the ice edge, up to 3 months earlier. I also wanted to look into the kind of importance of that bloom, that single bloom as a source of food to the system. And the way I did this was by calculating how much of the total production happened during the bloom as a function of the total production of the year. So a yellow value means that 90% of annual production is happening in one highly concentrated bloom, while the bluer colors indicate that it's more spread out through the year. And when we look at 1970, we can see that the Central Arctic has a really important bloom. Almost all the productivity for the year is concentrated in a single bloom. And that's less true when we go to like Washington and south of Norway over here. When we look forward to 2100, the Central Arctic remains a really important, still a really important single bloom. But at lower latitudes, it seems like maybe more and more production is happening outside of that single bloom. And that's particularly true in these two areas, which are two places where water is flowing into the Arctic. So overall, we're seeing this production less concentrated in a single bloom, especially in these waters flowing into the Arctic, which is important because phytoplankton are the base of this food web. And if production is more spread out of the year, it may represent less of a good food source to zooplankton and other animals that Anna just demonstrated for us. Okay, so this is some work on the distant future, but what about right now? We know the Arctic's already changing really substantially. A study by Mathieu Ardina used satellite remote sensing to try to understand current changes in the timing of phytoplankton blooms. And he divided satellite observations into kind of 3 different bloom types. He considered if there was no real increase in or change in biomass over time, he considered that to be a flat bloom. A single bloom has a single increase and a single decrease in biomass, the kind of classic phytoplankton bloom of the Arctic I've talked about. And then a double bloom involves 2 separate increases and decreases, one in the spring and one in the fall. And when he looks across the Arctic, he finds that that flat kind of style of bloom is most concentrated closest to the North Pole. And then he gets a single bloom, a little bit in a ring further to the south of that, and a double bloom outside of that. But he actually finds that the fall blooms, these double bloom features, are observed more and more frequently over time, and that a lot of the increase in their frequency is observed at higher latitudes. So I think overall, the story of light in the Arctic is one of consensus. Scientists are largely in agreement that increases in light are leading to phytoplankton blooms that start earlier in the year, and also growth that's less concentrated in a single bloom and is more spread out. Okay, our second story is one of nutrients. And this is, I think, a story that's very different. It's a story of big scientific debate, and one that focuses on productivity, that metric of how much growth is happening over a whole year. So fundamentally, the availability of nutrients controls how much phytoplankton productivity there will be. If nutrients are in relatively small quantities, you're going to get a smaller phytoplankton bloom. You'll also probably get smaller species associated with that, and they are less nutritious for the ecosystem as a whole. And then when you get more nutrients, that can support a larger bloom and larger phytoplankton species. A study by William Lee at Fisheries and Oceans Canada tried to look in person at how some of the changes we're observing in the Arctic are affecting phytoplankton species. So he was looking at a particular place in the central Arctic, and he found that over time salinity was declining, as you mentioned earlier, and temperatures increasing. And he also observed that nutrients were found in lower and lower concentrations over time. So these surface waters of the central Arctic are becoming warmer and fresher and more nutrient depleted. And he was curious how that would impact phytoplankton. So he divided his observations into small phytoplankton, those less nutritious guys, and large phytoplankton. And he observes that over time, small phytoplankton are increasing while large phytoplankton are decreasing, indicating that these less nutritious phytoplankton are starting to dominate in the future Arctic. So these observations fit into kind of one possibility for the future of the Arctic with regards to nutrients. And this, before I fully explain the future, I should explain to you kind of how stratification works in the Arctic. In the Arctic, in the ocean, water is, the least dense waters sit on top of more and more dense water beneath. And over time, we are, as sea ice melts, we're adding more and more fresh water into the surface of the ocean. And that makes it harder for deeper water to be mixed into the surface ocean. And the problem is that nutrients are largely in that deep water. So these sets of observations by William Lee seem to indicate that there's more fresh water in the surface ocean, and that's leading to fewer nutrients and less production, as well as smaller species. So this is one possibility for the Arctic and nutrients in the future. But I think it's quite uncertain, and there's a study by Kate Lewis that finds quite a different potential future. So she used satellite remote sensing to look at net primary production over time, how much production by phytoplankton is happening. And she looked between 1998 and 2018 and found this really large increase in phytoplankton productivity over time, like 60% increase. So she was curious about what was driving that increase in productivity. She at first kind of assumed it was a light story. As you melt more ice, you get more open water area, and that means there's more area in which phytoplankton can grow, as observed by satellites. And that certainly seems accurate for the first part of the time series. Net primary production is increasing, and so is open water area. But between 2012 and 2018, open water area doesn't really increase while net primary production continues to increase. So she instead considered changes in phytoplankton biomass. Biomass, again, being how much phytoplankton there is on a given snapshot of time. And she found that that was increasing over time in the second part of this time series. And what that tells us is not that the area over which there are phytoplankton growing is changing, but rather that the number of phytoplankton, the amount of phytoplankton growing in any given area is increasing. And if that is happening, then that's kind of an indication that nutrients are increasing. So she was curious to understand where phytoplankton biomass might be increasing, this indication that there might be more nutrients in the system. She looked at biomass across the Arctic and saw that there were a couple places with really large increases that kind of explain a lot of the pan-Arctic increase. Some of these are places where water is flowing into the Arctic. So these are places where there might be an increase in nutrient supply coming into the region and allowing for more phytoplankton growth. And she also sees some places where wind is helping to mix deep water into the surface ocean. So I think overall, these studies kind of support a second future for the Arctic. And this idea is that as sea ice melts, it is putting in fresh water into the system. But the other thing it's doing is it's opening up more of the ocean surface to wind mixing. And that allows for more wind mixing, which allows for more nutrients to be brought up from the deep ocean. So she finds that potentially the future of the Arctic will have more nutrients and more production, which allow for larger phytoplankton species to grow. So these are kind of the two observations we have at the present, but what do we have? What is our idea of what the future of Arctic nutrient supply looks like? Well, Martin van Coppenol had a really nice modeling study in which he tries to understand how productivity might be changing in the future. So he used 3 different models and looked at them between 1900 and 2000. They had pretty similar rates of primary production over this time. When he looks into the future, the models have really different answers for how much primary production there will be. One model sees a decline by the end of the century, and 2 others see an increase, one a moderate increase and one a very large increase. So I think overall this demonstrates to us that the future of production in the Arctic is very uncertain. And the reason for that uncertainty is fundamentally because scientists are really uncertain about the future of nutrient supply in the Arctic and which of these 2 kind of futures will dominate. Models and observations both support both of these futures in the Arctic. And I think potentially nutrient supply may be really different by different regions. I've kind of presented a pan-Arctic story here, but perhaps the story is more regional. I'm really curious for you all to watch Anna and Alice's animation, and you'll get to see which of these possible futures they chose to represent in their animation. Okay, the final story I'll share with you today is one of temperature. And temperature is fundamentally a story of the limits of science, some of the ways that science is unable to give us full answers about what the future holds. And it's also a story of changing species. So this is, I'll talk for a moment about a study by Mary Louise Timmermans. She used satellite remote sensing to try to understand how sea surface temperatures were changing in the Arctic. I showed you before some big changes in air temperature, and not shockingly, that leads to some changes in the surface temperature of the ocean, where most of the Arctic is getting warmer. There have been quite a few studies on the biology of this kind of region of the Arctic, the Norwegian Arctic. So I'll show you now some interesting work on how these changes in temperature are impacting species there. And you'll note this is not the place with the largest temperature changes. It's just a place that has been well studied. Another remote sensing study by Greet Nukermans tried to assess if temperature changes might be driving changes in phytoplankton species. She looked at this region just north of Norway, and in 1998, she found that warm water had kind of incurred into this region from the Atlantic, and that's represented by this red line. And she also was able to look at phytoplankton species, and she found these temperate phytoplankton that are typically not observed in the Arctic, and that they kind of are correlated really nicely with this increase in temperature. So it seems like there's a link between temperature and phytoplankton species. When we looked forward to 2012, you'll see that warm waters incurred even further into the region, and they're really high biomass of these warm water phytoplankton. So this demonstrates to us that Atlantic phytoplankton species are expanding into the Arctic and kind of taking over an environment that was typically controlled largely by Arctic phytoplankton. A study by Randi Ingvaldsen was curious. She was curious to understand if these changes in phytoplankton might be affecting fish up the food web. She used observations in this same region to separate fish communities into either Atlantic-style fish in red, Arctic fish in blue, or a mix of the two communities in white. And she finds that in 2004, again, there was a fair amount of warm water intrusion into this area, but you still see kind of Arctic fish living all the way down to 72 degrees north, although there are a lot of Atlantic fish and mixed communities in between the two. By 2017, the warm water has really expanded far into the region, and there are very few kind of pure Arctic fish communities remaining. So this kind of shows us that southern parts of the Arctic are becoming less suitable environments for Arctic fish, which probably sounds not great. And I think one challenge with this observational work is it can't really tell us why. It could be that these fish are relying on phytoplankton that are not there anymore. It could be that they're just brought in by the warm water and are able to outcompete the Arctic fish. So it's a fantastic study, and it also has, it lacks some of the data we might want to know about. So overall, I think that this story of temperature tells us that scientists know that temperature is increasing over time, and that those temperatures seem to be associated with changes in the species of the Arctic. But I think the other thing that the temperature tells me is some of the limitations of science. I've shared with you a bunch of observational studies today, and those observations are limited in time and space. They can't really provide us with information about what the future is going to hold. And I've also shared with you some modeling studies today, and they're pretty good at replicating observed changes and projecting future possibilities, but they lack some information about the impacts of a changing environment on the whole ecosystem. And yeah, as a result, science kind of leaves us with some holes, some gaps in our ability to visualize the future and understand. And I think that's what's really wonderful about the animation that Anna and Alice worked on together, is that it fills in some of the gaps that science can't really answer for us. So with that, I'm going to have Anna come up here and talk to you more about the animation process.
So I want to share a little bit about the process of making the animation and making the music. And this is a process that really started with me learning about some of the science that Courtney just introduced you to in a really beautiful and clear way. And it started also with me having the opportunity to visit NCAR, hosted by Alice, and getting input from lots of scientists researching the polar regions. So it started with this scientific research, and the animation also incorporates imagery that's often produced for scientific publications. So Alice produced computer simulations of Arctic sea ice derived from CESM outputs, and you can see some of those outputs on the left, and then I stylized and incorporated those in the animation. Arctic Shifts also presents speculative futures about what will happen to the food chain, and that means making choices between possible futures that are currently under scientific debate, like the effect of changing access to nutrients on phytoplankton that Courtney shared, and also speculating about changes to the Arctic food chain that currently fall outside the scope of simulations. For example, the impact that phytoplankton changes will have on the zooplankton that eat them. So some of those links between the Earth system and the ecosystem have space for speculation. At the start of the project, alongside gathering these scientific references, I was gathering visual references. So that meant everything from looking at imagery of phytoplankton and microscopic creatures like these etchings by Ernst Haeckel from 1904, to looking at imagery of sea ice like from NASA Visible Earth, and also thinking about how artists represent different scales and use diagrammatic visual language. So everything from this print by Louise Bourgeois, Pinus sylvestris, to this amazing animation by Nieto, Swallow the Universe, that uses these different kinds of diagrammatic language to link different biological systems. I then developed style frames as a way to explore the look and feel of the animation. I used textures that try and capture a sense of ice, and digital watercolor techniques that try and bring us to the ocean. So the style of the animation has these color washes that are offset from line drawings. And this was another way to try and nod towards uncertainty, towards different possible futures, and try and nod towards this combination of diagrammatic and poetic styles. I worked with two wonderful animators on the project, Nika Harrison and Suzanne Matarin, and they joined at the stage of revising storyboards and animatics. So this is where we work through the story beats and compositions. And at this stage, we were simultaneously developing animation tests. So that's where we work out the quality of movement for different scenes. So much of the movement within Arctic Shifts is based on reference. One of the aspects of the project where reference was really hard to find was depicting the growth of phytoplankton at the microscopic scale. So there are lots of photographic images of phytoplankton, but there isn't really any footage that shows how phytoplankton divide. How do you take a thing that's encased in glass and then multiply and divide? And one of the only depictions I could find of this stage of phytoplankton growth was from this book from 1910, from a 1907 expedition to the Arctic. And this book includes 36 plates, 2 of which are shown here. And here you can see an enlargement of the depiction of cell division of a centric diatom that served as reference for phytoplankton growth in the animation. So we can see this glass shell dividing in 2, and then the 2 hemispheres creating their own new glass shell. In terms of techniques, the animation combines digital hand-drawn frame-by-frame animation done in Adobe Photoshop with keyframe animation done in Adobe After Effects. And the animation kind of moves between animation that's based on reference to more exaggerated and sometimes playful movement, like the way that each creature eats. It's not based on reference. So I want to share a little bit about the process of making the music. Much of the music for Arctic Shifts was created using an algorithmic approach to music composition I developed with my dad, who's also here in the audience. And this is a process called gene network music. So with this approach, I create music that is modeled on biological processes. It's modeled on the dynamic interactions between genes within an individual organism. So through Arctic Shifts, I'm trying to depict interactions between creatures in an ecosystem visually, and then orally, through sound, I'm depicting the interactions between genes within an individual organism. So diatoms, phytoplankton, have roughly 10,000 protein-coding genes. Humans and polar bears have roughly 20,000 protein-coding genes. And these genes interact in a regulatory network, in a gene regulatory network, or gene network for short. So this network is what determines what proteins get produced where. So even though your entire genome is within each cell, it is through the interactions of the gene network that you get eye proteins produced in the eye instead of on a fin, for example, or you get stomach proteins produced in your stomach. So here is a visualization of a simple gene network. Here we have 7 genes instead of 20,000 genes. So the 7 blue dots represent the 7 genes, and the lines with arrows and plungers between the dots represent the interactions between genes. So you can see that gene 1, when gene 1 is activated, or another way of saying activated, gene 1 is on, or gene 1 is expressed to form a protein. When gene 1 is activated, that will promote the activation of gene 2. When gene 2 is activated, that will promote the activation of genes 4 and 6. When gene 3 is activated, that will inhibit or prevent the activation of genes 2 and 5. So what happens when these interactions within this network unfold over time? Well, at any given point in time, certain genes will be turned on, they will be active, while other genes will be inactive or off. And so we see in this example that the network actually cycles through 3 different states. 1, 1, 2, 3, 1, 2, 3, 1, 2, 3. So we get a kind of 3-beat pattern, a kind of waltz that emerges from these interactions. And so we see a musical potential here. So in creating gene network music, I simulate the gene networks in MATLAB. Here you can see a screenshot of some of the code. And I create patterns of gene expression over time. So here is a look at gene expression over time for a gene network with 170 genes. And the different colors just represent subnetworks that have higher levels of interconnectivity. And what we see here is that you start to see some patterns emerge. And you can imagine reading this gene expression over time as a kind of musical score, if you imagine mapping different genes to different instruments or different pitches. So with Arctic shifts, I use Logic as a digital audio workstation where I sonify or make audible the gene network music score. So here you can see a screenshot of the music for Arctic shifts being assembled in Logic. On the left are different tracks that correspond to the different instruments and sounds associated with each organism and element of the Earth system. So humans have a breath sound, fish have a flute sound, zooplankton have a string instrument sound. And at the right, you can see a zoomed-in view of one section of a phytoplankton track, where you can see notes that have been created using gene network music that are mapped onto a custom digital instrument that I created with Logic using sounds of class.
Let's just go ahead and give them a round of applause again for pushing over. And before you all leave, I just have a couple of little housekeeping things, and then I'll let you go, I promise here. Thank you all again for attending this lecture. We hope to see you again online or hopefully in person, July 16th from 7 p.m. for our next event. We'll chat with Mike Wiltberger and Ed Otten about space weather, how it forms, and why it matters to modern technology. If you're interested in more NSF NCAR Explorer Series events, check out our website for upcoming lectures and conversations and to review recordings of past events. If you are 18 years or older, which I see a lot of folks are, please take a moment to fill out our 3 to 5 minute anonymous survey to help us better understand the impact of this program and how we can improve for our future event. The survey will close next Monday, and we're going to pop up a QR code, and we have other QR codes, and we'll send you a reminder email. So please, please, please fill out that survey. I hope to see you all next time. With that, and I will also say any folks that need assistance on our stairs on our mezzanine, we will have a staff member that has a ramp that can assist you to use a ramp if needed. But with that, thank you all and hope to see you next time.